Aim: to create a realistic model of meiosis which will predict variations in genotype when offspring is undertaking meiosis. Materials: 1 coloured paper, 5 colours, 1 scissors, 1 packet of pipe cleaners, 1 pencil.
The first phase of meiosis is prophase 1. During this phase, DNA condenses and coils around each other to form chromosomes, maternal and paternal chromosomes. These maternal and paternal chromosomes then come together to form a homologous pair. The DNA in each individual maternal and paternal chromosome then duplicates, and the chromosomes split into chromatids joined to the centrome. Crossing over of genetic material then occurs. The two chromosomes in a homologous pair exchange corresponding alleles by breaking down at corresponding points in each sister chromatid. The genetic material is then exchanged, therefore introducing variations within a homologous pair, allowing greater genetic diversity. The nuclear membrane then begins to break down. A spindle apparatus begins to form and the microtubules attach to the centromes to the chromosomes. Metaphase 1 then begins. All chromosomes line up in pairs at the equator of the cell. Independent assortment is used to ensure that not all maternal and paternal chromosomes will be separated into different cells. Next is anaphase 1. The chromosome pairs are separated and are pulled into separate poles of the cell by the microtubules still attached to the centrosomes. Now that homologous pairs are separated, telophase can begin. The cell begins to expand and then divide into two daughter cells which are still attached at this point. The nuclear envelopes are formed again. Next, during the phase of cytokinesis 1, the daughter cells are now separated and are neither identical to each other or the original parent cell. The two daughter cells have half the amount of chromosomes of the parent cell. The second stage of meiosis, meiosis 2, now begins. During prophase 2, the chromosomes further condense and the nuclear membrane begins to break down for the second time. Spindle apparatuses appear again and the microtubules attach to the centromes of the chromosomes. Now metaphase 2 begins. And like metaphase 1, the chromosomes align in the equator of the cell attached to the microtubules. Anaphase 2 now occurs. The sister chromatids are pulled apart from the microtubules, pull them to opposite ends of the cell. The cells expand as the chromatids are now pulled away from each other. Next is telophase, when nuclear membranes are formed again and new daughter cells are still attached. Finally, cytokinesis 2 occurs. The four haploid daughter cells separate and are now genetically different from each other and the original parent cell.